Two, here we go. So let's learn a little bit about Bacillus thuringiensis. This is a different kind of bacterium, but this is one of the, we take one gene from this bacterium and it ends up being put into lots of different crops. So if you hear about Bt transgenic crops, they're going to have Bacillus thuringiensis proteins in there, and the proteins from that are going to be called cry proteins, CRY. Let's watch for a little bit, and then we'll pause this thing, and I'll go. I've got a couple pictures of cry proteins up on the other screen. I'll show you.
Side Information Center, here to talk with you about Bacillus thuringiensis, which is also known as Bt. We'll talk about what it is, how it works, and its toxicity. Bt is actually a group of closely related soil microbes. Each type of Bt makes a different kind of protein that is toxic to specific groups of insects. When these insects eat the protein, it changes shape and attaches to the insect gut wall, creating holes in it. Soon after, these insect larvae stop feeding and they eventually die. In contrast, when people eat the same proteins, they don't change shape and they don't cause us harm. Bt is found in over 180 pest control products. These products generally contain a mixture of Bt toxins. Varieties are only effective for certain types of insects. For example, Bt kurostaki products are used on garden plants to control caterpillars. Products with Bt israelensis are used around water to control immature flies and mosquitoes. Additionally, some plants such as corn, cotton, and potatoes have been engineered to produce their own Bt toxins. This is done by copying certain pieces of DNA from Bt and putting them into a plant's DNA. Plants use this DNA, like an instruction manual, to produce Bt toxins within its own tissues. That way, when insect larvae eat the plant tissues, they're exposed to the toxins. This form of genetic change in plants can reduce overall pesticide spraying to control insects. And when people eat these plants, the toxins do not work on us or cause us harm. In the environment, when Bt and its toxins land on surfaces, sunlight breaks them down quickly. They are also broken down by other microbes in the soil. However, breakdown is generally slower below the soil surface, where it's dark. In soil, Bt typically stays in the top several inches. There, it remains dormant until extra nutrients become available. In water, Bt is not known to reproduce.
And ad break is a good place for us to take a break. That's actually at a pretty good spot. If you like, you can go on and read more about this. Look for the Kurzgesagt, Kurzgesagt, which is German for in a nutshell, or basically briefly said, I think. And Kurzgesagt GMOs. And it's a nice little, nice little video on these and kind of how they work. And I like that one. That's kind of cute. Okay, so let's go and look at some cry proteins. So here is a cry protein. Let's get this thing three-dimensional. Here we go. And we've always got to make it spin. There we go. All right. So there's our cry protein. And so you can see we've got three real domains here. We've got a mostly beta strands domain down at the bottom. We've got a second beta strand domain, this sort of red one here. And we've got a long alpha helix domain up at the top. I think it's the alpha helix domain that changes shape and actually binds to the guts and becomes like a membrane pore. But I'd have to look that up and make sure. That may not be known, or, well, it may be known, but I sure don't know it. How's that? Yeah, there we go. That's a cry protein. Over here is a group of actually three of those cry proteins. These guys are assembled to make one of those little pores. So let's look at that. Here we are. Naturally, we got to spin this bad boy. And so there they are. Once they're transformed, they're all interacting with each other. And you've got a little membrane hole right there, right down the center. There we go. Right there. Boop. And that's where, the guts can basically leak out of the the inside of cells that are lining the intestines of insects. And that's how these proteins seem to work. They clump together. They only do this in the presence of certain proteins from the insects. And again, as far as I'm aware, we don't know the name exactly of those proteins, but I don't know. Eventually they'll figure them out. So there you go. That's level 2.